Now, again, this is the most basic part of your biological basis. We'll start with neuron, the structure of neuron, and all of that kind of thing. Very basic, very simple. So please pay attention, make notes as much as possible. Uh, so the nervous system, the most basic block of the nervous system is the neuron, right? There are more than 100 billion neurons in the human brain, and they transmit information electrochemically, right? So there is electricity and there are chemical molecules that are there that transmit information on and on and on. So for me to say all of this to you, there are millions of neurons just going on and on, right? There's electricity running and chemicals and neurotransmitters, synapses, all of that is just flowing out and all of that information is coming out to you, right? There are sensory neurons, motor neurons, everything is going on at once. Now, this is what a neuron looks like. We are all familiar with this. But the three most major parts of a nucleus of a neuron is your this cell body, right, or what is known as the soma. Then you have dendrites, right, and then you have this thing called the axon, right? These are three major parts. The cell body is this one. Um, it contains the nucleus, right, which contains the DNA, which again is the most basic thing in terms of creation of the human being and having information passing on and detailing how your growth should take place, right? So the DNA is the most basic thing. From the DNA, you get the neuron, and from the neuron, you get the entire nervous system and the rest of your body, right? Within the nucleus, you also have the endoplasmic reticulum and the ribosomes. Both of these are responsible for creation of proteins and synthesis of proteins. And there is mitochondria, which all of us know is the powerhouse of the cell, right? It's the most important energy center. So the mitochondria will synthesize the proteins and then there'll be energy released and the energy will help the neuron pass electrical current, right? If this dies, the entire neuron is absolutely useless. Now, the second part is the axon, right? This part, this yellow thing here. The yellow thing is actually not the axon, it's the purple thing, but it's lined with myelin sheet. Um, this is a cable-like projection of the cell which helps transmit information, right? It carries a nerve impulse or what is known as the action potential along the length of the cell, right? Now, sometimes the axon is covered with myelin sheet, sometimes it's not. So it's not necessary that all axons have to be covered with the myelin sheet, right? Uh, the myelin sheet is basically fat which covers the axon and it helps pass electricity faster, right? And it also insulates it, so it helps it from getting damaged. Now, in the peripheral nervous system, right? The peripheral nervous system is the nervous system that is not your brain and your spinal cord, right? So it's in the periphery of your body. The peripheral nervous system has myelinated neurons, right? So there is this myth that all the neurons in the brain have myelin, not really true, right? Very few neurons in the brain have myelin covering it, right? Whereas there are more myelinated neurons in the peripheral nervous system. So the nervous system that is not your brain or your spinal cord, right? That emerges out of your spinal cord like that. That is your peripheral nervous system, uh, which has both sensory and motor neurons. Now, there are more non-myelinated neurons in the brain and spinal cord, right? Now, another thing that is always told to us is that the myelin sheet helps in passing of electrical stimulation quickly, right? That is true. However, the other way it also works is that in between the myelin sheet, if you see here, these purple gaps, these are known as node of Ranviers, okay? So sometimes in some neurons, what happens is electrical current will come in, action potential will take place, and electricity will literally jump between these nodes of neurons, which makes it even faster. So the myelin is not even necessary to pass electricity, right? That's the job of the node of Ranvier. So sometimes they also help in passing electricity quickly, right? Um, then you have your third part, which is the dendrite. These are your nerve endings, right? They create connections with other cells. So the other dendrite or the postsynaptic neuron will be here of the other cell. And dendrites are like tree-like structures, right? You can have multiple dendrites for one neuron. You can have so many different branches that are going out, right? And they can be located on both ends. So the dendrite can be here, and in the axon terminal, it can be here also, right? And it's just a space where it's like a courier thing. It just transfers information, right? Um, the axon terminal, which is this part, that is the end of the neuron, right? This is where the neurotransmitters are held, right? It's somewhat enlarged and it looks like a golf club. It's a button shape, like a button mushroom. Imagine a button mushroom. That's what it looks like. And it has the neurotransmitters within it. So when electrical current passes through the neuron, it stimulates the axon terminals, which release the neurotransmitters into the synapse, right? So they make synaptic connections with another nerve cell or effector cell. An effector cell is a muscle cell or gland cell, right? Or a nerve cell is basically another neuron. Uh, and in that gap, right, between the presynaptic neuron and the postsynaptic neuron, that place is called the synapse, right? Um, and the nerve impulse will come in, it will release neurotransmitters will be in that gap, and then it will get sucked in by the receptors of the postsynaptic neuron, right? Sometimes they will close, sometimes they'll open up. We'll discuss that later, right? And synapses are also of two types, actually, right? This is again one of the favorite questions of net because it's not taught in depth. There are chemical synapses and there are electrical synapses. So the synaptic cleft, right? That gap is known as the synaptic cleft. In that space, sometimes it will hold chemical currents only, right? Chemical neurotransmitters only. Sometimes that cleft will only hold ions, right? Those are electrical molecules, basically. Chemistry, if you go back, there are ions and anions, right? So ions are there. There'll usually be sodium, potassium, or calcium ions that will be there, right? IONS. So there are two types of synapses, actually. The chemical synapses are where your neurotransmitters are there. So dopamine and serotonin and etc. are chemical synapses. Electrical synapses are ions, right? Some synaptic clefts have both. So they'll have both ion, electrical synapse and chemical synapses, which is the most common one actually, right? Now, neurons also classified into multiple ways. Okay, one is the basis of differentiating neurons is the kind of work that they do, right? So that's known as the functional classification. What kind of work does the neuron do decides its functional classification, right? This is something we are all familiar with. There are sensory neurons, motor neurons, and interneurons. So the sensory neurons carry input messages from sense organs to the brain or the spinal cord. So sense organs to the central nervous system is sensory nervous system, sensory neurons, right? So sense organs are obviously your taste, smell, sight, hearing, right? So those input messages from your body basically into your brain and spinal cord is sensory neuron. Motor neuron are output messages from the brain or the spinal cord to the rest of the body, right? Usually all information goes to the brain except when you're having a reflex action. So if you touch something really hot, your body will immediately remove the hand, right? That's basically the reflex arc that is controlled by the spinal cord. Apart from that, most things are just controlled by the brain, right? Uh, then there are interneurons which connect the sensory and the motor neuron. So sensory information comes into the brain, it's linked with the interneuron, which then passes the information to the motor neuron, right? So they connect basically the entire nervous system with each other's working. Very important neurons to have. Then there are glial cells. These are cells that don't have anything to do with electrical stimulation. These are just structures that hold the nerves together, right? They hold the neurons together, and they're very, very important because glial cells, once they start to die out, the neurons will start to die because they will start to hit each other, right? And then they'll start to damage one another, and therefore you will start to develop multiple forms of mental illnesses. So glial cells very important. They hold the entire nervous system together. The Schwann cells that I was telling you about, these are present in the peripheral nervous system, right? So the glial cells that are present in the peripheral nervous system are known as Schwann cells. In the central nervous system, there are four different types of glial cells, right? Four different types of glial cells. The most prominent one are oligodendrocytes, then there are astrocytes, then there are epidermal cells, and there are microglia. Now, if you just remember oligodendrocytes for the net exam, it's enough because they very rarely mention the other three. But if you remember all four, that's good for you, right? I would suggest you remember it as OAEM, right? And then make an acronym that helps you remember, right? But just remember, Schwann cells are the only ones that is for the peripheral nervous system. Any other of the other four is for the central nervous system, glial cells, right? Glial cells are what? Things that hold the neurons together and their entire surrounding area. So inside the brain, very important. They are like branches, like trees. They keep everything in place, right? Then there are structural classifications of neurons. So these are how the neuron looks. So functional classification is in terms of what the neuron does. The structural classification is in terms of what the neuron looks like, right? So you have anasonic neurons. These are basically neurons that you cannot differentiate between the axon, dendrite, cell body, your myelin sheath, nothing. You can't differentiate between anything, right? They're small, they have no clues to distinguish dendrites from axons, and all cell processes look alike. So if you can see that image, right, you can realize that it looks like a place where you don't know what is going on, right? Anytime there can be electrical impulses going in any direction. And therefore we don't really understand them very well, right? They're mostly located in the brain and some sense organs that are mostly related to your smell and sight, right? Um, but we really don't understand them because there is, like, if you look at it under microscope, it'll just be like constantly getting stimulated and you don't know what stimulation is going from where. Otherwise, normally there is a certain pattern, right? But here it'll just be moving in all directions at the same time. So very difficult to understand. Then you have bipolar neurons. Here the, no, the cell body is located at the center of the neuron, and then axons are on both sides, dendrites are on both sides, right? Bipolar neurons are also rare. They are also present in certain sense organs where they relay information about sight, smell and hearing from receptor cells to other neurons, right? So it's sight, smell, hearing for your bipolar neuron. So touch is not there, right? And what is the fifth one? Fifth sense organ is what? Sight, smell, taste, right? Taste and your tactile sensation, these two do not have bipolar cells. Bipolar meaning two, bi meaning two, right? And two sense organs are not there. What are those two sense organs? Touch and taste, right? So if you remember it as bi TT, right, or bit, that's how you remember that what bipolar cell neurons are not present in, right? And they're not present in touch and taste, right? They're smaller than unipolar or multipolar neurons, and they measure less than 30 mm from end to end. Then you have a unipolar neuron, right? A unipolar neuron looks very similar to a bipolar neuron, if you notice, except that the cell body is protruding out, right? The cell body is protruding out of the entire system, right? It's also sometimes known as the pseudo unipolar neuron. So the dendrites and axons are continuous or fused, and the cell body lies off to in the center at some point. In one, it's there outside the cell basically, right? So it might be on one end, the other end, or it'll just be outside. Um, so in such a neuron, the initial segment lies where the dendroid is, right? This is again present in the peripheral nervous system. Then you have multipolar neurons. Multipolar neurons have, oh, I think I've mixed the pictures up. Yeah, I think I mixed the pictures up. Just ignore the pictures. Multipolar neuron is this actually, right? This is a multipolar neuron. I don't know why that's written there. They have more than two dendrites and a single axon. This is what is mostly seen in the entire central nervous system, where you have a cell body and you have multiple dendrites, right? And then you have one long axon and then again multiple dendrites. Now these are usually present for to control skeletal muscles, right? What are skeletal muscles? Skeletal muscles are all the muscles in the body or organs that are voluntarily controlled. So your entire muscular system is usually skeletal muscles. Things like your heart, your liver, etc., are smooth muscles, right? And this is the most widely present form of neuron because if you think of it evolutionarily, this makes sense, right? To have dendrites in multiple places, having multiple connections with other dendrites who also have multiple connections, right? And to have the energy center on one side. So it makes a lot of sense to have more multiple polar neurons throughout the nervous system. Okay, then you have nerve conduction. This is very important. Luigi Galvani, right? Write down this name. This question might come, right? Saying who was the first person to discover electrical charges in the brain or in the muscles, right? So the guy's name is Luigi Galvani. Um, so he found that the neurons are capable of generating electrical signals, right? And they pass information between each other using those electrical signals. So this was a new fascinating sort of insight, right? And then when they dug deeper into it, they started to realize all these things about action potential and synaptic transmission and all of that, right? So what he basically figured out was that in a neuron, right? If you look at a neuron, outside the neuron, there is something known as the int- interstitial fluid, and then inside it is the axoplasm, right? So axon, axo, that's the fluid inside, and then there you have the fluid outside, right? Whenever there is a potential action potential taking place, there are places that open up that pass electricity from outside the neuron to inside the neuron, which is what allows the entire action potential to flow through, right? And these concentration of charged particles are again known as ions, right? So you have sodium ion, potassium ion, and sometimes there are calcium ions, right? So there is a electrical potential difference between what is inside the neuron and what is outside the neuron, right? So sometimes outside the neuron is negatively charged and inside the neuron is positively charged, right? Inside the neuron is negatively charged, outside the neuron is positively charged. So when an action potential takes place, there are openings that occur which allows the positive energy to come inside. It depolarizes the neuron and it allows the action potential to flow through, right? So action potential, these are the things you need to remember, right? Point one is the point of resting membrane potential, right, which is minus 65 or minus 70 millivolts, right? So this is a point where the neuron is ready to fire, but there is no electrical stimulation, so it's just resting, right? Now, if an excitation comes in, sensation comes in, then it will start to depolarize, right? It will depolarize, the sodium channels will open and the membrane will depolarize and hit point 2, that is minus 55 millivolts, right? This is the threshold of excitation, or what is known as the depolarization point, right? And then the stimulation will go on, right? Your potassium and your sodium ions will go up and down and inside out, and the potential will keep getting positively charged till it hits 30, right? At which point the action potential leaves, and then the stimulation has passed through the axon. Then the sodium channels will close, the potassium ions will keep leaving, right? And because the positive charge is leaving the cell, it will start to repolarize and it will go down, and then it will keep going down. At point 4, you have reached hyperpolarization. Here, so much potassium has left the cell that the cell will enter into the refractory period where it will not get stimulated and cannot fire. Then slowly again the potassium will start to enter the cell, right? It will create the positive charge in the cell, sort of positive charge, slightly higher than below minus 70, and then it will come back to minus 70, minus 65, and then it will be again in the state of resting potential, right? And then this will keep going on and on and on and on as long as the cell is continually being utilized, right? Relatively straightforward. You don't need to remember whether your calcium goes in or potassium goes out or sodium goes in, or at which point potassium goes in and at which point it goes out. None of that is necessary. Just remember the points, right? Minus 70, minus 55, plus 30, and below minus 70, right? What each of those things mean, and know that sodium is positive, potassium is positive, calcium is negative. If you know that much, you are sorted. Then that is not the only kind of transmission. This transmission, action potential, is within the neuron, right? One neuron is there, it has gotten information from the other neuron saying you are stimulated, now move. So it will move, it will pass the electrical current through itself, and then by the time it passes through the axon, it will hit the terminal buttons which will open up the neurotransmitters, right? From that point onwards, synaptic transmission will take place. So this is a synaptic transmission, right? Action potential has taken place. It has opened all these terminal buttons or axon terminals, right? Which will then release the neurotransmitters, right, into the synapse or the synaptic cleft, this space in between, right? These neurotransmitters will release, and then they will go and bind themselves to the postsynaptic neuron, right? This is basically the dendrite of another neuron. These neurotransmitters, when they attach themselves to the postsynaptic neuron or the dendrite of the next neuron, it will stimulate that neuron, and then another action potential will take place, and then that action potential will release another few neurotransmitters which will attach to the next neuron, which will again create action potential, and so on and on and on and on. Your body keeps functioning, right? Your brain keeps functioning. So action potential is within the neuron, synapse is in between the neuron, right? Sometimes what happens in the synapse is when you have, say, drugs or alcohol and all of that, right? Even medication, even pain relief, what's happening is it works in multiple ways. In one way, the action potential takes place, right? And an excess amount of neurotransmitters is secreted, right? Normally, when you get happy, a certain number of dopamine is released, for example, right? But if you, say, have some drug which releases more dopamine,
Then when you have that drug, say marijuana for example, you have marijuana, right? It will release extra dopamine from your neurons, from the neurotransmitters, right? Which means there is more dopamine that is being secreted into the next cell, which means that you will stay happy for longer, right? Same thing if you have an hallucinogen, you will start hallucinating for longer, right? Uh, so that's one way in which there is excess neurotransmitter that is released. Another way is they will release the same amount, but they'll block the excess amount from coming back. So one of the features that is there is that if you have some good incident and you get happy, right? Suppose 10 neurotransmitters release, okay? There are 10 neurotransmitters in your terminal button, but your happiness level is of only 5. Electrical current will come in, it might release 6 or 7, right? But then you're supposed to get happy only for 5. So 5 will attach to the next one, and then these two will be sucked back by the previous neuron, because your happiness level is only to be for 5 neurotransmitters, right? But when you have drugs or any form of illicit substance, or any substance that is not required by your brain, right? Um, Sometimes they'll release, if there are 5 required, they'll release 10, 15 like that. And they will be there in the synapse, and each time they will keep reinforcing the next neuron to get more happy, more happy, more happy, right? Or what they'll do is they'll release 7 and they'll stop the blocking of the other 2 extra ones. So you get happy for a little longer amount of time, right? So that's called reuptake. The reuptake gets blocked. Now, usually one of the other mechanisms that is present is that if there are excess neurotransmitters that are not required, they start to disintegrate also in the synapse, right? Your external substances, even medication, stops that disintegration, right? So those neurotransmitters work like that basically in between the synapses. Sometimes there'll be excess release, which will stimulate the next neuron for a longer amount of time. So you'll stay happy or sad or whatever for a longer amount of time. Sometimes they'll block the reuptake, right? If you say depression medication, for example, they are usually serotonin reuptake inhibitors. So they will stop the serotonin that is released from being taken back. So serotonin will stay in the synapse for longer. It will keep attaching to the next neuron for a longer amount of time, and it will keep stimulating it for a longer amount of time, which will keep making the person more excited for a longer amount of time. So depression levels will start to dip. So basically what's happening is you have an action potential that goes through the neuron. It reaches the terminal buttons or the seminal vesicles or whatever the different names for it. They release neurotransmitters. These neurotransmitters then move into the synapse, and then they attach to the next neuron, the dendrites of the next neuron, which is also called postsynaptic neuron. When they attach to the next neuron, they stimulate that neuron, and then that neuron will have action potential, and then it will release neurotransmitter, which will then stimulate the next neuron, and then so on and so on and so on, right? Now these neurotransmitters, when they bind to a postsynaptic neuron, when they bind, they don't stay there forever, right? They bind and they stay there for a while, and then they, once their use is gone, they disintegrate, right? And then you stop feeling whatever. Suppose you have a moment of, say, happiness, right? One neurotransmitter is released, it goes and attaches to the next neurotransmitter. Suppose you're supposed to smile only for 2 seconds, it will give only that instruction, say 2 seconds smile. Once 2 seconds smile is done, it will disintegrate, right? So similarly, right, multiple neurotransmitters are released and they attach to the next neuron. Now when you intake any sort of substance which is not natural for your brain, including medication, they work in 2 ways. One is, if there are, say, 5 neurotransmitters that should be secreted for a particular behavior, when you have that substance, it might release more, it might release 10 of them. So when 5 will attach to the next one and disintegrate, there will be 5 more in the synapse to attach, right? So you'll stay in that state for a longer amount of time. So when people have, say, marijuana is one classic example, right? Or when they have alcohol, they stay sad or they get angry or they get happy, and it's slightly longer. You notice that people start to laugh a lot more, right? They will be inhibited for a longer amount of time than normal. That's because there are more neurotransmitters there which are binding to the next receptor, right? So that's one way in which there is extra release of neurotransmitters. Another way is the uptake that is there. Like suppose 5 are required, they release 7, 5 go and bind to the next one. There are 2 extra neurotransmitters that are remaining, right? Those 2 are usually taken back by the previous neuron, but some medication, some drugs, whatever, they stop this reuptake. So 5 will attach, and then there'll be 2 more left to attach, right? So you'll stay in that state for a longer amount of time. So reuptaking is inhibited, as well as releasing of neurotransmitters is much more.